Okay, good morning, everybody. Let's get going. Um, so I think we finished the field utilization unit 1, right? Mostly, but probably just adipose tissue left. Um, so let's. So basically, we talk about the different tissues. They have a specific um, utilization of the different metabolic fuels, right? Oh, and um, oh, sorry. Okay. Kind of slow. Okay, so now let's just talk about adipose tissue. So what are kind of fuels that adipose tissue use, and primarily fuels that adipose tissue can use, um, glucose and triglycerol, and fuels that you released into the circulation from adipose tissue, including the free fatty acids. Basically, it's from the hydrolysis of triglyceride, which stored in the adipose tissue in the fat condition, right? So release of fatty acids mostly occurs in the fasting condition, um, and glycerol, right, is also released from, again, the breakdown of triglyceride. And this is an important enzyme, remember HSL, uh, inside of the adipose cells or skeletal muscle cells. And HSL basically hydrolyze triglyceride and release fatty acids, glycerol. But we also mentioned, if you still remember, in addition to HSL, there are also other important two enzymes, like lipase. One is called ATGL, adipose tissue triglyceride lipase, and the monoacylglycerol lipase, right? And three different enzymes and catalyze different locations of the fatty acids from glycerol backbone and release fatty acids. So these three lipases work together to completely break down the triglyceride and release three fatty acids, molecules, and glycerol. And lactate can also be released from adipose tissue from the partial oxidation of glucose. Again, adipose tissue compared to red blood cells and muscle, adipose tissue is not a major contributor of a lactate, right? But adipose tissue does release the lactate. Um, so the two actually major things that release from adipose tissue during starvation, fatty acids and the glycerol, right? And glycerol can be picked up by liver and to synthesize the glucose, right, through gluconeogenesis during starvation. Um, so this is basically what kind of fuels that and adipose tissue used and released from during the starvation. And we all know that the primary role of adipose tissue is to store and fatty acids in forms of a triglyceride and also release fatty acids, right, depend on the physiological state. And in the fat state, we know that the primary role of adipose tissue is to pick up fatty acids from triglyceride into main the lipoproteins, right? Still remember the two main lipoproteins that transport the triglyceride from diet, from small intestine to liver, right? Chylomicron and VLDL, right? And the enzyme hydrolyze the triglyceride within these two lipoproteins, LPL, right? Lipoprotein lipase, which is released from adipose tissue during the fat condition, and insulin stimulates, right, release of LPL. And in the fasted state, and which enzymes break down triglyceride into monoacylglycerol and 3 fatty acids. Remember, we just talked about it. Right? HSL, yes, HSL is the enzyme that activated during the starvation and hydrolyze triglyceride within the adipose tissue and release fatty acids into the circulation. Okay. Okay, now let's talk about the unit 2. And in the unit 2, we are going to talk about specific the role of adipose tissue in fuel metabolism. I mentioned earlier the adipose tissue. Well, everybody knows, right? Adipose tissue is the main function of adipose tissue is to store energy, store triglyceride. But in addition to this function, adipose tissue has other very important role in regulation of the whole body metabolism. Because adipose tissue now recently, since I think the 1994, when the hormone that was discovered secreted from adipose tissue, then adipose tissue now is considered as an important endocrine organ, meaning that adipose tissue can release or secrete the very important hormones and peptides, right? So the first hormone that was discovered from adipose tissue is leptin. So we're going to talk about the metabolic role of adipose tissue. So let's review this again. We know that protein cannot be converted to carbohydrate, right? Oops, sorry. So both carbohydrate and protein can be converted to fat, right? Which you all agree, right? You know, the acetyl-CoA is a key molecule that can be generated from oxidation of carbohydrate and protein, and acetyl-CoA is the substrate or precursors for fatty acid synthesis, right? So both carbohydrate and protein, if we have too much in the diet and body cannot use all of them, then they have to be converted to fat and stored in the adipose tissue as the triglyceride. So, and we also know that fat cannot be converted back to protein or carbohydrate, right? If we have too much fat stored, can we? No, right? Except for two specific type of fat, remember, which two? Glycerol and propionyl-CoA, right? And in general, fat cannot be converted back to carbohydrate and proteins, right? So what are we going to do with extra fat? You know, how we get rid of the extra fat if we have too much, you know? Burn it, right? I already show you. So burn it. So we have this is probably is the most, only way, I mean, our body deal with the extra fat, right? We burn it. But what kind of, the metabolic pathway that you're thinking we can burn fat? What's that? Yeah, lipolysis is one of the pathway, but fat means fatty acids, right? Lipolysis, we just try to break down triglycerides and release fatty acids. But what are we going to do to get rid of the, yes?
You want to activate the enzymes that breaks down fat, so that instead of exercise, the HSL.
Yeah, right? So HSL to break down triglyceride, right?
Yeah.
And release fatty acids. And what is the next step? What are we going to do with the fatty acids? What do we, how we get rid of? Yes, beta oxidation, right? So fatty acid oxidation, beta oxidation, so then we can completely oxidize fatty acids, right, to get rid of extra fat. So this fatty acids oxidation is very important functions or pathways in our body. We try to get rid of the fat. So anything with, wrong with the fatty acids oxidation, the most common reason or defect is the mitochondrial dysfunction. It's also very common in the type 2 diabetic patient. The mitochondrial function is reduced, particularly in the muscle, so then cannot efficiently oxidize fatty acids, right? Can efficiently burn, fat. So this is something, it's kind of, pathological conditions. Now let's talk about obesity and metabolic syndrome. Obesity is really a epidemic, and it's kind of balanced, right? The energy intake, energy expenditure need to be balanced. Is a major contributor for the obesity development is the imbalance between energy intake and energy expenditure. Mostly we either eat too much or we lack of exercise and we burn less of the fat or energy. So this imbalance is a major contributor in causing the development of obesity. And very unfortunately, in the United States, they're also leading the obesity prevalence. Obesity rate is actually the highest in the United States, like 31% of population. Our population as our overweight or obese is really is the problem. And not just obesity, but obesity very closely correlated with a number of metabolic syndrome, right, like cardiovascular disease, insulin resistance, type 2 diabetes, all related, even some type of cancers like colon cancer or breast cancer, they're all related to obesity. And this just show you the map, the growing, the rate of obesity is going so fast. If you compare to 1990, we were kind of very skinny, right? But now you see that almost all the states in the USA, the rate of obesity goes up a lot, right? During the last decades. And actually, the nation, like NIH, invests a lot of money and actually even increase the research money to study obesity, to try to control obesity, but it seems not so efficient. I mean, spend money, but still have no clue to control obesity. So body mass index is the body weight versus the height and square. So we would like to have a desired BMI, which is between 20 and 25, right? Desirable range. But if BMI is higher then 25 or between 25 and 30 consider the overweight, and 30 to 40 is obesity, and greater than 40 is severe obesity, right? So the BMI is usually people use to define the obesity. And also, you probably also notice that the gender difference in terms of the body composition, the fat versus water. Actually, men seems to have a high percentage of water and less percentage of fat as compared to women. Right? Women seems to have a higher percentage of body fat. And the distribution of fat is actually more important than the total amount of fat that we have. For example, the abdominal obesity, sometimes also called apple obesity, means that you have actual fat accumulated in the abdominal region. And this type of obesity most common in men and is also more close related with the metabolic syndrome, right? So abdominal obesity seems to have a higher risk for the development of the metabolic syndrome. And the other type of also called a female pattern of peach, the obesity. That means that fat they accumulate mostly in the hip and side region, right? And this type of obesity is less metabolic harmful compared to abdominal obesity. So if we look at a little bit more detailed, the body fat distribution is more important. The upper body fat distribution, or also called visceral obesity, as I mentioned, is closely correlated with the cardiovascular disease, insulin-resistant type 2 diabetes, and we usually use the waist to hip ratio to predict the risk of the metabolic syndrome. For example, if the waist to hip ratio is greater than 0.85 for women, or greater than 0.95 for men, this will consider is high risk. If you have this higher ratio, then you will high risk for the development of a metabolic syndrome. And the other important thing I wanted to mention is called ectopic fat deposition. So we know that most of fat we wanted them to wanted to be stored in the adipose tissue, right? So adipose tissue is the right location for the fat storage. And some actually in very severe obesity patient or type 2 diabetic patient, for some reason their adipose tissue has a dysfunction. They cannot actually store the properly, the triglyceride. So then the triglyceride cannot be properly stored in adipose tissue and goes to the other tissue stored in the other non-adipose tissue. For example, liver, right? So causing a lot of fat accumulate in the liver, causing fatty liver disease. And also you have actual fat accumulating in the heart or skeletal muscle, right? In the obesity. So this kind of a situation called the ectopic fat deposition. It's pretty common in the severe obese patient and type 2 diabetic patient. Okay, now what is the metabolic function of white adipose tissue? We know that store lipids, right, and maintain the lipid homeostasis is the main important function of adipose tissue. And adipose tissue is also important to regulate glucose homeostasis. For example, GLUT4 expression level in adipose tissue is high, and adipose tissue is one of the tissue that take up insulin stimulate glucose, right? Glucose uptake along with the skeletal muscle. So it's also an important tissue to regulate glucose homeostasis or glucose levels control. And the third important thing I just mentioned, that endocrine function, right? So the adipose tissue can produce a number of peptides, hormones, cytokines, right? And regulates the whole body energy metabolism. And I just wanted to show you adipocyte. You see it's very different from the other type of cells. Not only the size is bigger, but it's the structure of the cells is also very different, right? It's a huge lipid droplets, I feel with huge lipid droplets, and the nucleus actually push to the edge of the cells, and very little the cytoplasm region, right? The adipose cells. And this is the real adipocyte. You look at under a microscope, right? It's very different from other type. You almost see the nucleus, right? So it looks like it's just a bubble and oil. So filled with the large lipid droplets. And functions. We know that adipocyte is insulin responsive cells, means that they have a insulin receptors, right? So insulin can bind these receptors, and one of the important function is the glucose uptake, right? And GLUT4 is the transporters that regulated by insulin and increase the glucose uptake. And the second important thing is that the adipose cells can also respond to insulin and release LPL, which can hydrolyze the triglyceride in combined quantity VLDL and release fatty acids and pick up by adipocyte and store as the triglyceride in adipocyte, right? And adipocytes also respond to the catecholamines and have adrenergic receptors, right? And respond to the, for example, catecholamine, epinephrine or not epinephrine, and during the fasting, and catecholamines release increase, right? And can bind this receptor and activate HSL. Remember HSL function or activation, and then HSL hydrolyze the triglyceride inside of cells and release fatty acids and glycerol into circulation during the starvation conditions, right? So adipocyte plays a very important role in terms of lipid metabolism in fat and fasting condition, right? Fat condition, you will respond to insulin, and fasting condition respond to the catecholamines. And adipose cells also release a lot of interesting hormones, right? And like leptin, I mentioned, is the first hormone that was discovered, released from adipocyte in 1994, and then later on we discovered other the hormones that also released from adipocyte. For example, adiponectin, resistin, and in addition to this important cytokines or adipokines, the adipocyte also release the other cytokines, right? So here I just wanted to focusing on the two important and adipocyte-derived hormones, leptin and adiponectin, how these two adipokines or hormones regulate the energy metabolism, right? Fuel metabolism. So first one, leptin. Leptin is released from adipose tissue, right, and has a central and peripheral targets, meaning that leptin released into circulation can go to the brain and regulates appetite, energy expenditure, and also can go to the muscle and regulates fat oxidation, right? So basically leptin is a good hormones, right? Increase the energy expenditure, decrease food intake, so allow, tell us to eat less and burn more fat, right? The two main target tissues, brain and muscle for fat oxidation, brain for food, suppress the food intake, right? Any questions? Right. So in terms of regulation, the leptin is fasting for 24 hours, then can cause the decrease the leptin. So it means that during the fasting, leptin production is reduced, which makes sense, right? The fasting condition, we don't want to burn fat, right? We want to save, energy. So fasting, during the fasting, leptin production is decreased. And if we refeed animal, which is actually results from the animal study. So if we refeed animals, then the leptin production will increase again very quickly, within like 3 to 6 hours, and leptin productions increase again. So insulin also regulates leptin production. So question here, why does the body makes less leptin during fasting? I just talked, right? Wanted to save energy, right? Leptin basically is a burn fat, right, to waste energy. There are interesting mouse models, the OBOB mice is actually leptin deficiency. That these mice, they don't have leptin, so you can compare to the normal mice. The size is double, very obese, and they don't have leptin, so they eat a lot, because leptin, we know, is to suppress food intake, right? And these mice, they don't have leptin, so they cannot control eating behavior, so they eat a lot. And if we inject the leptin to this OBOB mice, and can reduce the body weight and can rescue the obese phenotype, right? They reduce body weight and reduce the food intake. So it's very interesting. And the other type of model is called a DBDB mice. The DBDB mice basically is a leptin receptor deficiency. They still have a leptin levels, but even higher than the normal mice. But the leptin has to bind to receptor to function, right? So and this DBDB mice, they don't have leptin receptors. So although they have a high level of leptin, but the leptin cannot function. So they're similar the phenotype. They also eat a lot and also gain a lot of weight and very diabetic. And this, what do you think if we inject leptin to this DBD mice? Will leptin will work? No, right? So leptin will not work in the DBDB mice because of lack of leptin receptors. Okay, so now let's talk about the edible nectin. Is another adipocyte-derived hormones, the adiponectin, is also secreted from adipose tissue, and adiponectin has the two peripheral tissue targets, right? They don't have a central effect, meaning that they don't have a effect on the brain, but the two main targets of the adiponectin are liver and skeletal muscle. They act to the liver and the skeletal muscle and increase the fat oxidation, right? So adiponectin is also considered the good hormone, right? It can stimulate, fat oxidation, can help burn a fat and maintain the body weight. It's also synthesized from the adipose tissue, and it also actually decreased during the starvation. Similar reason, right? The adiponectin is also increased fat oxidation. We don't want to burn too much fat during fasting. So fasting condition, adiponectin levels also goes down, and refeeding also quickly restore the adiponectin production from adipose tissue. And insulin also can stimulate the adiponectin gene expression and production. And in the type 2 diabetes patient, insulin resistant conditions, the plasma adiponectin actually levels decrease, right? So which may be related to the less efficiency of the fatty acid oxidation in the type 2 diabetic patient, right? They have a less leptin, less adiponectin, so they can efficiently burn the fat in the obese and type 2 diabetes. And here I wanted to talk about a little bit more sort of a molecular mechanism, how leptin and adiponectin stimulate fat oxidation, right? So this is a general pathway. Try to remember this AMPK. We mentioned this kinase before, right? So AMPK is the important enzyme that can activated by leptin and adiponectin. And AMPK is the key enzymes that can control fat oxidation, right? And so we will go to the detail, how AMPK regulates fat oxidation. But AMPK is a target enzyme by leptin and adiponectin, right? So adiponectin activate AMPK and then increase fat oxidation, right? So AMPK is the serum athreonine kinase, right? It's also considered a few sensing enzymes. That means the AMPK activity can be regulated depend on the energy status inside of cells. So it can be activated when the energy level is low, right? When ATP level is low in the cells, so then can activate AMPK, then can stimulate fat oxidation and try to produce more ATPs, right? So again, the energy status, particularly the low energy levels, can stimulate AMPK and stimulate fat oxidation. And the consequences of the AMPK activation is to increase both fat oxidation and glucose oxidation, right? So once you activate AMPK, then both fat oxidation and glucose oxidation will be all increased. And again, the purpose is try to make more ATPs, right? So AMPK can be activated during the starvation, right? Starvation, a lot of cells actually have a lower ATPs, and so then AMPK can be activated and then stimulate fat oxidation. Okay, so this slide I think is important, right? Just give you a clear picture how leptin, adiponectin, active AMPK, and fat oxidation, right? So leptin and adiponectin binds to receptors on the cell surface and activate AMPK, right? The stimulate AMPK phosphorylation activates it. And AMPK is the important enzyme to catalyze the ACC. Remember ACC, right? So ACC phosphorylation. So once ACC is phosphorylated and its activity actually decreases. So in other words, that AMPK is activated and decrease ACC activities, right? And ACC is the key enzyme for this reaction, right? For catalyze the formation of marino-CoA from acetyl-CoA. Remember this, right? And marino-CoA again is a key molecules, a substrates that can inhibit CPT1 on the mitochondrial surface, right? Membrane, and CPT1 is the key enzyme that controls fatty acyl-CoA transported from cytosol into mitochondria for beta oxidation. Right? Is that clear, this pathway? Right? Again, the leptin and adiponectin stimulates AMPK activation, right? And AMPK, once it's activated, and decrease ACC activities and decrease the formation of marino-CoA, right? What does marino-CoA do for the CPT1? Remember, you guys? Inhibits, right? So marino-CoA inhibits CPT1. So when marino-CoA levels goes down, this inhibition will be relieved, right? So then CPT1 activity was increased when marino-CoA levels decrease. Does that make sense? Right? So then beta oxidation will be increased. Yes?
So the receptors on the cell, what kind of, is this still an adipose tissue or just a general cell?
Oh, this is the general. I mean, in the skeletal muscle,
Oh.
I mean, also have the AMPK, and liver, a lot of cells actually have AMPK, have these pathways. So liver muscle is a major pathway, right, for adiponectin and leptin mainly in the muscle cells.
So they have receptors.
Right, receptors on the liver and skeletal muscle, right? Yes?
So there's leptin, adiponectin, phosphorylate AMPK, which activates it, which phosphorylates ACC, which deactivates.
Correct. Yes, yes. So again, AMPK, once it gets phosphorylated and activities increase, right? And ACC, if it's phosphorylated, the activities decrease, right? It's different. So AMPK phosphorylate ACC and decreases activity, right? And decrease production of marino-CoA and increase CPT1 activities, right? Does that make sense? Okay. And here I wanted to remind you the glucose transporter in skeletal muscle, it's sort of alternative mechanism. We know that skeletal muscle, the glucose uptake is mostly occur in the insulin stimulated condition, right? The GLUT4 is the main glucose transporter in adipose tissue. So in response to insulin, GLUT4 goes to the cell surface and pick up or transport the glucose, right, from circulation into the muscle cells. And there is also an alternative mechanism that can also drive GLUT4 to the cell surface, which is independent of insulin. and we mentioned this before, remember, right? And so the exercise can increase the GLUT4 go to the cell surface and transport glucose, which is again independent of insulin. Even when you don't have a high level of insulin, exercise itself can drive GLUT4 to the cell surface. And the mechanism actually is here. So muscle contraction during exercise, muscle contraction can activate AMPK, right? So AMPK then drives the GLUT4 to the cell surface, right? This is represents the cell surface membrane, plasma membrane, right? Drive GLUT4 to the cell surface and pick up glucose, right? So again, the skeletal muscle, the pick up glucose independent of the alternative mechanism that independent of insulin, this through the AMPK activation, right? Exercise activates AMPK and then stimulate GLUT4 go to the cell surface and for glucose uptake.
This only happens in muscle.
Yes, this is a muscle. All right. I think I will finish this, and I'm going to talk about the Unit 3, right, and 4. We're going to talk a little bit about the metabolic fate of micronutrients in response to food intake and metabolic fuel utilization. So in this 3 and 4 unit, we're basically focusing on the different. We divided the stage, the different stage depend on the fat fasting condition, right? The more precisely, divided stage based on the time. For example, the absorptive phase, which means that 2 to 3 hours after meal, and then post-absorptive phase that start from 3 to 4 hours after meal till several days of starvation, right? And then we'll talk about each phase and how our body uses the different fuels or switch the fuels. And then also we're going to talk about the metabolic fuel switch, means that, for example, in the fat condition, we usually use the dietary sources of energy, right? And then during the starvation, we basically use the endogenous fuels, either from the stored energy or synthesized fuels. So and also the other switch is from the utilizing more carbohydrate during the fat condition and switch to the utilizing more fat during the fasting condition, and how this switch being regulated, right? So let's talk about the metabolic fate of the fuel metabolism in response to food intake. So I also mentioned that the carbohydrate, protein, and fat metabolism in a different physiological state, and here we more precisely divided the phase based on the time, right? For example, absorptive phase, or also considered fat condition, which means that 2 to 3 hours after meal, and this period of time we usually consider as a fat condition, right? Fat state. And the post-absorptive phase or fasting condition means that 3 to 4 hours after meal, 2 days of starvation. So normally when we eat after 2 or 3 or 4 hours, we start to eat second meal, right? But if you don't eat second meal, and this start from that time, and then actually you sort of enter the starvation stage. And the second thing that we're going to talk about is how does the body switch metabolic fuels during starvation? we will discuss a sort of series of random cycles. And the last thing is how does the body knows when to switch metabolic fuels, right? For example, I mentioned switch from endogenous, using exogenous or dietary sources to the endogenous fuels, or switch from using carbohydrate to using the fat as the energy fuels, right, during the starvation. And this regulation of fuel switch basically by a lot of hormones, substrates, enzymes, right, transporters, right. So standard meals, we usually in our meals we have about 90 grams of carbohydrate, 30 grams of protein, to 20 grams of fat, which count for like 27% of the calories, right, from the 20 grams of fat. And so how our body distributed or metabolized this dietary carbohydrate, protein, or fat, in the absorptive phase, like 2 to 3 hours after meals. So the absorptive phase, right? So carbohydrate 90 grams, glucose in the meal. Usually we know that our body digests the carbohydrate and pick up the carbohydrate, right? And first of all in the enterocyte in small intestine, and by this glucose transporters, right? This SGL T1 is the glucose transporters located in the enterocytes, right? And pick up the glucose. And then the enterocytes, once the glucose enters enterocytes utilize some of the glucose, right, as energy fuels, and then the rest of the glucose being absorbed into the body and leaves the enterocyte through this transporter, group 2, if you still remember. So high glucose levels will occur after meal, and then this will stimulate insulin release, right? You remember this, the whole process in the absorptive phase. So I guess, let's see, maybe talk one more slide. So once the glucose being absorbed into the body, the liver takes up 20% of the absorbed dietary glucose, right, by this glucose transporter, group 2. And glucose in the majority of the glucose in the liver is actually go to the glycogen synthesis pathway, right, in the fat condition. And some of the glucose, they actually go to the partial oxidation and to produce the lactate, and this lactate being used for gluconeogenesis. And the gluconeogenesis, though, again, can also occur in the fat condition, right? Depend on the tissue. And the glucose once is, and the purpose of gluconeogenesis in fat condition is try to recycle and conserve the glucose carbon skeleton, right? So once this glucose is produced and is usually stored in the glycogen in the liver in fat condition, right? So again, in the liver, liver metabolizes 20% of glucose from the absorption, right, from the diet, and the majority of the glucose in the liver being converted to glycogen and stored in the liver. So glycogen synthesis in the liver, the glucose can come from the two sources. One is the directly from the dietary glucose, right? And the other sources is through the gluconeogenesis pathway from precursors, lactate, which initially from the partial oxidation of glucose, and also amino acids in the fat condition can also be used as a precursor for gluconeogenesis. So all these two precursors, lactate amino acids, can be used to make a glucose, and this glucose, the purpose of this glucose is being stored in the liver as a glycogen, right? Okay, so I guess I'm going to stop here. Let's take about 10 minutes break, right? And then we have a quiz.